So, we are going to discuss granulomatous inflammation now. So, first of all, we will see the definition of granulomatous inflammation, and this definition is also from the Robbins textbook. Granulomatous inflammation is a form of chronic inflammation. This is the first thing that you have to notice. It is characterized by the following things: collection of activated macrophages, which we also called epithelioid cells, often with T lymphocytes and sometimes associated with necrosis. So it can be said that granulomatous inflammation is a form of chronic inflammation which is characterized by activated macrophages, often with T lymphocyte, with or without associated necrosis. So this is the breakdown of the definition. Now we will study about the pathogenesis of how granulomatous inflammation takes place. So this is the diagram from the Robbins textbook, but I have simplified it for you because it might be a little tricky for you to understand. So here I've made an easy diagram for your better understanding. The first thing is a very basic thing which you need to know is that if any antigen enters our body, it is directly not attacked by our immune system. It is basically first processed and then presented to our T cells or our T lymphocytes so that it can attack the pathogen. You can compare it to like you can't eat food if you are not if it is not served in a platter. So you can think that the antigen presenting cell, these are the antigen presenting cell, this cell basically take the antigen and put it on a plate to our T cells so that they can act and help in the immune response. So this antigen presenting cell concept is really important. Antigen presenting cell can be many cells can act as antigen presenting cell. There can be professional antigen presenting cells. There can be non-professional antigen presenting cells. For example, I'm writing some name. A macrophage is an APC, and also, for example, a dendritic cell. So these are two examples of APC or antigen presenting cells. So as you can see, the antigen, this antigen entered the cell here, got processed, and then was served on the plate. What is the plate? The plate is basically an MHC. Now, at this moment, if you don't know what MHC is, that is not relevant here. Just think of it as the plate or the receptor through which the antigen is presented. Presented to whom? It is presented to my CD4TH1 cell. CD4TH1 cell. This CD4TH1 cell also has a counterpart. It has a counterpart, a T cell receptor, which helps in detection of this antigen that is presented through my uh, MHC molecule and the antigen presenting cell. Now, once this antigen presentation is done, then a few things can happen. The first thing that can happen is this CD4TH cell can release cytokine. This is the first thing that can happen. Can release cytokine like tumor necrosis factor, which helps in recruitment of leukocytes. You can see this is a blood vessel, as we discussed previously also. From the circulation, the leukocytes enter into the tissue. Here, leukocytes are also known as WBC in case you didn't know. It's really common knowledge, but still I'm mentioning. So, here, in case of granulomatous inflammation, the principal leukocyte that we take is basically a monocyte. Why? Because monocyte is the name of this cell in periphery, but when it enters in the tissue, this monocyte, when it enters in the tissue, we call it a macrophage. So this the recruitment of the leukocyte takes place. The macrophage enters from the blood circulation into the tissue. After entering, the CD4 Th helper cell also releases interferon gamma. Now, this is an important molecule because this interferon gamma causes this macrophage to get converted into an activated macrophage or an epithelioid cell. As we have already discussed in the definition, that an activated macrophage or an epithelioid cell is a very important part of granulomatous inflammation. Now, you have to know what are the differences, the main differences between a macrophage and an activated macrophage or an epithelioid cell. So just from the diagram, you can see that a macrophage is a round to oval in shape, and it is an eccentric nucleus. That means the nucleus is in the periphery, not in the center. Whereas in an epithelioid cell, you can see the shape is described as elongated, or it has a famous name, slipper-like, the one you wear on the foot, slipper-like cell with an central nucleus. Here I would also like to mention the difference between epithelial cell versus epithelioid cell. A epithelial cell is basically, it doesn't have a basement membrane to hold its cells. But in epithelial cell, there is a basement membrane. For example, if this is the basement membrane, on this we can have the epithelial cells. But in epithelial cell, there will be discrete cells, but there will be no basement membrane to support them. It will be absent. Okay, so this was uh, this much, and then we till now considered two factors. The first one was release of cytokines, which helped in recruitment of leukocytes, and then the second factor was the release of interferon gamma, which results in activation of the macrophages. Now, these activated macrophages do two things. Basically, I am studying chronic inflammation. Chronic inflammation means anything that is chronic, I would indicate, or I would say that it's ongoing, right? So if basically if the activated macrophage would just eliminate the pathogen or the source of the injury, then it would have been over. There was no, chance of chronicity, developing a chronicity. But that doesn't happen. So one thing you need to remember, that in general chronic inflammation always happens in response to a pathogen that is difficult to eliminate. So, since it is difficult to eliminate, what happens is my immune response starts increasing. Since the pathogen can't be eliminated, my immune response keeps increasing, and it keeps increasing so much so that instead of damaging the pathogen, it starts damaging my normal tissue. So, now this is a harmful condition. So, in that case, the body does uh, a certain thing, which is it tries to contain the infection rather than trying to remove the offending agent. So that is why chronic inflammation takes place. Moving forward, we saw the both one and two things that happen. The third thing which happened is how the activated macrophage or the epithelial cell acts. It acts by two mechanisms. First of all, I've already said you that a macrophage is also an antigen presenting cell. So the activated macrophage of the epithelial cell can itself cause antigen presentation to the CD4 Th cell. So this is one. And another thing that can happen is the same cell can release interleukin 12 that helps in proliferation of T cells. So this whole process, I would say that the formation of epithelial cell and the activation of T lymphocyte by interleukin 12 or by antigen presenting cell by antigen presentation through the epithelioid cell leads to formation of granuloma. This is what happens in granulomatous inflammation. Now, what is a granuloma? How is it formed? We've already studied. And here it's the same thing, it's just a pictorial representation. This is the picture of a granuloma. So, what are its components? What are the different things we are going to learn in the next video? So, this is this much for today.